polariton in plasma molecule scaffolds.
Yes, thank you. Thank you for the invitation. I am a few hundred meters away from the Institute of Physics. Although I am myself a physicist, so I feel very comfortable talking here. And what I want to tell you about in thinking about the topic of the conference is how we try to take quantum phenomena and reproduce it in molecules and in nanoparticles and in general things that are floating in solution. So I think that is the key aspect that I'm going to show you now is taking electronic coherence processes and then putting them in a MOG and seeing if they can survive there. So I'm going to be basing the talk on something called polaritons, but instead of doing them in a cavity, we're going to do them in nanoparticles. So polaritons are these hybrid-like matter states that exist when we put molecules inside two mirrors. So there is somewhere an electronic transition that couples strongly to the mode of the light confined in the cavity, and then we can get strong coupling, we can get upper and lower polariton branches. So this is also something that can exist in infrared, as was presented in the talk before mine. And this is, for example, what Giuseppe Ferruccio does here. But there's another way of doing them. If you take, instead of a mirror, you take a plasmonic nanoparticle and you have a surface, you can have a very, very small cavity. And this allows you to have strong coupling, but with a small amount of molecules. And you can go even further. You can just remove the surface and you can have only the nanoparticle, and the plasmonic mode is going to act as an open cavity. And so you're going to get polariton states, but with the possibility of coupling it to a molecule. So as I said, what I want to talk about in this talk is how do you make systems clean enough, in particular because I'm going to be talking about nanoparticles and molecules floating around in solution, how do you make them clean enough to control quantum mechanical effects? And maybe the other way around, these are complicated systems, so how can we measure the photophysics in detail? And thinking that once you have a system, you want to use it for something, and before you use it for something, if it's an excited state, you want to understand the photophysics. So once you do, you maybe can think about any applications. So I'm going to talk first about, as an introduction, the topic of plexitons, or polaritons in plasmonic cavities. It's called a plexiton because it's a plasmon plus an exciton. And I'm going to tell you about, mostly about structural characterization that my group has been doing in a prototypical system. It sounds like a little bit nitty-gritty, but I want to convince you that this is what you need to do if you want to build complex materials that have really a chance of turning into quantum technologies. I'm going to mention very briefly work that we've done on another type of nanoparticle, cesium lead bromide. Then I'm going to finish by talking about spectroscopic protocols that we have been developing in my group in collaboration with Sweden. Okay, so let's start with plexitons. What we have two ingredients. We're going to have a plasmonic transition. This happens in a metallic nanoparticle about hundreds of nanometers wide. Then we have some type of molecular transition. So here is a very broad plasmon resonance. We have a very short molecular transition. When we put them together, in this picture, you have an upper and a lower polariton branch. These are samples that we make in my group. And you have a lot of possibilities to play with them. You have, as a first ingredient, the plasmonic nanoparticle that can come in a variety of size and shapes. And typically, you are going to choose very ugly shapes. So the most successful ones, we don't make them in my group, but they are called nano urchins. So it's just this horrible thing with spikes, because you can concentrate the field there very strongly. And then you have also a complication of what you choose as an emitter, and I'm going to talk only about molecules. And when you enter the world of molecules, you have a lot of possibilities. So let's say you can understand what I'm going to say thinking about a collection of two-level systems. So you have a molecule, there's an electronic transition, it's a two-level system. You put it in a solution, it changes a little bit, and then you start to couple it to other molecules around it. And you are, most of the cases, having a Coulombic interaction between the two, a dipole-dipole interaction. And depending on the angle of this interaction, you can couple, have a negative or a positive coupling, which means that your bright state, the one that is going to be called the exciton state of the aggregate, is going to be changing position from a J aggregate to an H aggregate, and then you have some disorder here. And you're going to have also a manifold of dark states that you are not going to see in the absorption spectrum, and their position is also going to change quite a bit. So again, depending on the molecule you have, you put it in solution, it aggregates in many different types of ways, and this, you're going to couple it to the plasmonic cavity. So there's a lot of complexity here. And this is just a review that people from Italy did on the kinds of plexitons that appear in the literature. And you can see that now there are hundreds of systems that are being reported in the literature. And this comes from the ability to mix a metal, usually gold or silver, in a specific shape and a molecule. And with all of them, you get a different type of photophysics. So there are, again, three, I would say, four ingredients. You have a nanoparticle. This nanoparticle in chemistry, we use some type of molecule to protect it. It's called the capping layer. And then you have the molecule that is going to couple to the dipolar mold of the metal. And then there's going to be a solvent, and sometimes things are going to be moving around. So if in the best case scenario, you get a coupling, you have the cavity mode, the quantum emitter, and you split into an upper and a lower polariton cavity. But as I mentioned, there are many degrees of freedom here. Inside the metal, you have an electron reservoir. Outside, you have molecules, but you also have states that are dark, and they're going to play a role in the photophysics. And then you have the solvent that is making things come apart. And in general, this is a cartoon, you can have at least 4 types of interactions. You can have a molecule sitting on top of the metal, you can have it linked by a molecule, you can have it floating around in solution, being electrostatically attracted to the capping layer, or you can have something where it's mixed. So a lot of possibilities. And this is not work from my lab, but also from Italy, where they do experiments and they model it by having a molecule sitting on top of the capping layer, sitting flat on the surface, or even acting as a ligand between two particles. Okay, so it's not so simple. You have many options that are happening in solution, and sometimes you don't even notice what is happening. So you need to do a thorough characterization. And this is what my group is doing. If I were to mention something about the photophysics, I can start about talking about cavities. So if you put some type of emitter inside the cavity, in this case this is LH2, a photosynthetic system, and you measure the lifetime. If you measure it without the cavity, you have a decay here in gray. And if you put it in the cavity, you have an increase in the lifetime. So this is good. You have an excited state, you put it in the cavity, you make it live longer, you're going in the right direction. And this happens because you suppress radiative transitions. But if you look at it in the cavity, in the molecular system, let me see if I will show it with the laser pointer. If you look at the molecule that is a J aggregate, you have a decay that looks like this, and then this lives for a very long time, about nanoseconds. It's a very long-lived state. And the moment you put it inside or on top of the nanoparticle, you have this black line here that almost goes to zero, but it goes very fast. So you go from a few nanoseconds to 3 picoseconds, which is the opposite of what you want. And we studied this system very thoroughly, and what happens is that you have charge transfer pathways that come because the molecule is sitting right on top of your cavity. And this doesn't happen when you have fabric cavities. So that's a problem. In general, cavity increases the excited state lifetime. That's good, but something else happens when you have a nanoparticle. So there are a few problems. Nonetheless, it's a good idea to try and investigate polaritons in solution because you have them in the liquid, so you have the possibility of the polariton state interacting with molecules in solution and doing some chemistry. And this is why we are interested in. So the geometry plays a crucial role. If you think of, for example, nanoprisms that we also have looked at, you have emitters or molecules that are sitting here. The coupling, the dipolar coupling happens at hotspots, so only these molecules and you have a lot of stuff that is not playing any role in the absorption spectrum, then you have a big electron reservoir here, you have the possibility of transferring charge, and the point is that you need to understand this mess well enough in order to make the materials better. And I will give you two instances where we're trying to make them better. But, so just 3 points. The additional degrees of freedom in the Plexiton are going to dictate the dynamics. In particular here, when you excite the molecules, they're going to quench via the surface. These are charge transfer pathways. And all the photophysics has to be very, very sensitive to the geometry. So what is the wish list of my lab? That we can get better at making these systems so that we can direct where the energy is going. So something that people can do in the cavities is they can put donor and acceptor, and they can put energy transfer from one to the other via the cavity. In principle, we could also do this if we learn how to put two different types of molecules together. The second thing, which is kind of the reciprocal of this, is you don't focus on the molecules assisted by the cavity, but you look at the plasmonic cavity assisted by the molecules. These are some calculations by a group also in Sweden, where there is a small metallic cluster and a molecule next to it. And what they find is that when you excite the joint system, you end up generating hot electrons, and these hot electrons occur at the energies of the upper and the lower polarity directions. So the take-home message is that whenever you have a metal, you excite it generates hot electron holes. It has its own properties. It happens at the energy of the plasmon, but when you have upper and lower polarity states, you generate them at the energies of the molecule. So you can use the cavity to affect how the molecules are transferring the energy, but you can also use the molecule to affect how the electron reservoir is being excited. So these are all things that would be useful to understand if you know them, if you know the system a bit better. So we did what we were hoping not to have to do, which is looking at what the structure of this messy nanoparticle covered in a cyanide dye looks like. So this is a collaboration with people from Chalmers that do computation, people from France where we do the experiments, the transient absorption experiments, Giuseppe Niesenia for the Terahertz Raman experiments, my student Jorge, who is synthesizing a lot of nanoparticles, and Sinan Balci from Turkey that taught us how to synthesize. And this is really a prototypical system, and most of them will look like this. You have a chromophore. This is a conjugated electron system that absorbs in the around 600 nanometers, and then you have some little chains and anchoring groups that stick to the surface. Okay? So every Plexiton looks more or less like this. They have a strong J-aggregate transition, and they can bind to the surface. And our surprise was that when we do structural characterization of these dyes, there is very little in the literature. In fact, for this TDBC that is used in a lot of polaritonic chemistry, there was no study or very few studies on what the aggregate looks like. So this is one of them by the group of Bawendi at MIT. And what they do is they form an aggregate. So they take the molecules, they make them stack in a monolayer, and then they cap it with a silica film. And the way that they infer the geometry is they measure the height. And this is 2.7 nanometers that happens to correspond to a geometry where the sulfonate groups, so these are the sulfonate groups, they are alternating. Okay, so this is the geometry that they find. And what we set out to do was to understand the geometry of the Plexitons, but we ended up having to look at the geometry of these other components. So you can take the dye and look at it by itself in methanol, or you can look at it in water, where it will form aggregates, or you can look at it as Plexitons. These are the nanoparticles that are used to put the dye on top. And the combination that we started to use and we keep using in my lab is a mixture of density functional theory, NMR experiments, and Raman spectroscopy. And they turn out to be perfect to analyzing these disordered systems. So I'm just going to show you the idea of the different spectroscopic signatures that we're using. And first I tell you what are the stars. So when you have the molecule by itself, this is an absorption spectrum in thousands of wave numbers. You have a very broad absorption. When you bring them from methanol to water, they aggregate, they form these monolayers, and then you couple them to a plasmon and you generate the upper and the lower polariton branch. And they look like this, like really it's a suspension, it lives at room temperature, and they can be stable for several months in the fridge. And then we do DFT, we just take a bunch of different geometries, and we take them by themselves, we constrain sometimes geometry to be flat, we let them curve, we make them into dimers, we stick them on surfaces, this is what Joachim Halvin does. And then he calculates the Raman spectra. So this is the baseline from where we start. And then you can do experiments with NMR. So NMR is going to be a technique where you measure the chemical environment of spins, and depending on the type of molecule or the aggregation, you're going to see different Larmor resonances for the spins. Okay, so I don't explain this, I just show that when you have monomers or J aggregates, you can see them, they have different shapes, and there's a way to understand that. The most important thing is that there's a certain kind of experiments, they also kind of complexitons, a certain kind of experiments that is called noisy, where you can take two spins and you can make them interact, or they interact via a dipolar interaction, and this dipolar interaction can appear in a two-dimensional map. So in the previous talk, there was a discussion of cross peaks when the molecules are interacting. In this case, if you have two spins far away, you don't see any cross peaks between the two spins in this 2D map. When they come together, you start to see cross peaks. So it's very useful because you have the possibility of measuring distances in a system even though it is disordered. And we do that. I just show you the absence and the presence of cross peaks in this noisy experiment. So in the monomer, we don't have any peaks. These are different regions of the NMR spectrum. For example, if you look at this one, here I labeled them with these two resonances or these two resonances. And then this one in yellow is this one. So whenever they are in methanol, these ones are too far apart to see each other. And when you put them in water, they appear, which means that, I will show the geometry in the next slide, that they have to be in a certain geometry. And we can do this with several other cross peaks that appear, so it's just tracking the possible geometries with a model in the lab that we built, so we don't miss anything. And the conclusion from this is that we have a geometry where the sulfonate groups are upside down, so they are just alternating. And this is a boring conclusion, but it is a direct evidence that we see alternating sulfonate groups. This is consistent with what is hypothesized on surfaces, so that's fine. We're not going against what was measured in surfaces, even though sometimes the surface can template the atom. And then we start to look at the Raman spectra, and there's a lot of things that we can learn from the Raman. We measure the monomer, and we compare it with what has been done in the calculations, and we identify several key regions. And from this, we can conclude that we have a curved geometry. So this is just going through many modes of calculations, many simulations, and comparing them to the experiment. And we can do the same thing with the dimers, even though here the conclusions are a little bit less in agreement. We have the high frequency region that is in agreement, and then we have some stuff happening at low frequencies that we don't understand so much. But then we can do a correlation. So again, this is all very nitty-gritty work. Maybe it's a bit boring to explain it in the talk, but at least I convey the idea that you really look at all of the vibrational fingerprint of the monomer of the molecule when it is in the aggregate, and then you start to see what is happening when it is on the surface. And on the surface, we see three things. First, we see some exclusively plexitonic modes. So we see some vibrations in green that only appear in the plexiton, so they are good proxies whenever we start to modify the system. Then we also see some aggregation-sensitive modes. So these are these ones. They are absent in the monomer, but they are present in the plexiton, so we know that there is some stacking of the molecules on the surface as well. And then, strangely enough, we also see that some of these modes are monomeric in character. And the conclusion that we draw from this is that the aggregate is the molecule becomes an aggregate, sticks on the surface, and then there starts to be curvature on the surface. And this might seem like too much information, but when we are going to start to modify the system with different types of molecules, these are all the fingerprints that we need to collect in order to understand how the molecules are responding, for example, if you put a donor or an acceptor. We also started to look at this new spectroscopy for us. This is done in collaboration with a group of Giuseppe Peluccio and Yesenia, who's a postdoc in the group. And it's normal Raman, except that you look now at the terahertz region. And this terahertz region, it's great. I am preferring it to the normal Raman region because it becomes very sensitive when you have soft matter. So when you have molecular aggregates or when you have nanoparticles, you can see it in this spectrum. So for example, if you look at the monomer in red, you see a certain spectrum. When you look at it in water, you start to see order that appears. For example, you see this peak that is new. And when you see it in a plexiton, everything is, you see some redshifts in the peak. And the most striking thing that we've seen, that we don't understand, is that when we calculate the Raman cross-sections, the plexiton has a mode at 140 wave numbers that becomes enhanced by a factor of 40. So we don't know what that means, but it is something that I think will be useful in probing more complex systems. And we can understand what this mode is based on the FT calculations. Okay, so the conclusions is that if we want to understand the photophysics, we need to understand how the molecule is stuck on the surface, because you have these charge transfer pathways that are short-circuiting the excitation. That's not what you want. So you need to do a bit of relatively boring work to understand the structure before you can excite it. And we can, in fact, gain a lot of understanding. So we understand that the TDBC exists in a curved geometry in methanol. We understand that the aggregate is alternating sulfonate groups. So this is going to have an impact when you stick it on the surface. And we also know that when we absorb it, there's going to be an increased disorder of some of the chains. So this is now also very useful that we have identified certain modes that maybe once we understand them, that's fine, we understood the system, but once we start to modify it, we're going to keep a close look at these spectroscopic signatures and see how the molecular layer is being modified. So in terms of the wish list, now that we have finished this, we're going to try and make these types of hybrid donor acceptor systems. And this molecule, TDBC, you can get it from a company called Few Chemicals in Germany, but you cannot get it in Mexico. We tried for 1 year, it's impossible. So now we started to synthesize it. And I have never synthesized a molecule in my life, but this one will be the first. So I'm very excited to try something new. And once you know how to synthesize it, then we're going to make some modifications to get donor accept. And then we can also do, hopefully we can engineer the excitations in the metal. So we're doing transient absorption measurements to probe this in detail, but I'm not going to show it today. I do want to show another example where if you think any material that has interesting excitations, interesting optical properties, you can almost always port it to solution. But when you bring it to solution, it means you have to cut it into a nanoparticle, and you need to put some type of stabilizer, and this stabilizer is going to affect the photophysics a lot. And so Diego Solis is a researcher in material science, and we were wondering about understanding a bit better the emission in cesium lead bromide, which is also a prototypical system in emissive nanoparticles. So they look like this when they are illuminated. They have a very strong PL. They are very ugly cubes if you look at them in the TM, and you can change the molecules that are stabilizing the surface. And the idea is that people just put together ligands. So this ligand is positively charged, this one becomes negatively charged. They mix the types of fractions that you can get, and then they change the photoluminescence. And in fact, with NMR, you can look at this a lot more in detail. I'm not sure how much time. So I have 10 minutes, 15 minutes, 10 minutes. So we decided to look at this in more detail. And what you do is you take these little cubes in solution, they're functionalized with some ligand, in this case oleic acid and an amine, and you throw in another molecule. I'm not explaining what types of molecules, for now it doesn't matter, because I just want to talk about the spirit of doing NMR and understanding in a lot of detail about the photophysics. So you titrate, so you change the layer that is covering these nanoparticles, little by little, and you look at it with NMR. And again, NMR, you can take a look at all of these ligands, and because the way that the spins relax in the magnetic field, the line shape is going to change, the position is going to change, and you can learn whether they're adsorbed or not, but also whether they are moving or not. So in this case, I'm just going to show one of these resonances so you get an idea. If you look at an oleic acid resonance and you start to throw in this molecule, you're going to change the layer. And when you first start to throw in some molecule, you see that there is a set of range of concentrations where the shape becomes the same. And then at some point, it's going to change. So in particular, this shoulder disappears. These things becomes different and moves. And then you keep throwing in molecule, and you come up with a different NMR line shape. So by following this, you can infer the structure of the molecules that are passivating the surface. And you take the photoluminescence, you integrate it as a function of the molecule that you add, and then you compare to the regions obtained by NMR, and it turns out that they agree perfectly. So if you take, or as perfect as one can hope. You take the photoluminescence and you integrate it. This is what these dots are showing. That when you throw in this molecule, the photoluminescence increases, so you quench radiative pathways, and then you start to decrease, and then you start to decrease, but with a different slope. And in here, in these dotted lines, you have region A, B, and C. These are given by NMR, but they agree very well with the changes in photoluminescence. And if you spend a little bit of time thinking about the surface chemistry, you start to understand that if you have recombination sites, so at first you have bromine recombination sites, then the photoluminescence goes up, you quench this relaxation pathway, then you keep adding more molecule, you open lead recombination sites, and then the nanoparticle degrades. So again, this is all, the details are all in the paper, but what I want to mention is that really, if you understand the ligand layer quantitatively, then you can start to understand what type of electronic structure defects are driving the recombination, and you can make the materials better. And this is just something that we're doing in my group right now as a precursor to studying the photophysics. So this is the little cartoon that I used to explain in a longer presentation, where you have something that looks like an ugly bug, but you see here it looks like Lego pieces. So if you can identify what are the Lego pieces, then you can put it all together. Right. So this is kind of working with the dirty part of the systems, of the molecules, of the nanoparticles, but we're also very much interested in novel probing protocols to characterize the excitation. So I'm going to tell you something that is on the other side, which is the cleanest thing that you can think of, which is atoms. And this is done, this work that I'm going to present is done with Professor André in Lund, Hugo Laurel, and David Busto, who were in Lund, and then Jorge Baños, who is a student of mine. And Anton Frisk is a co-author in one of the papers, although I'm not going to present that one. Okay, so let's think that we have this ugly metal, it's covered with molecules. We want to understand the photophysics. We do a typical transient absorption spectrum. So we come in with a pump, we excite, we wait a little bit of time, and then we come in with a probe. And there are many flavors of these kind of experiments that you can do. You can have an experiment where you do transient absorption. So this will be a pulse that can be a white light, so you can probe a broad region of the spectrum, or you can do a single color, it can be lower noise, or you can do what was presented in the previous talk, two-dimensional spectroscopy. And this is problematic when you're measuring these types of molecular systems, because what you have is a non-equilibrium electron distribution, for example, inside the metal, and what the light probes is an energy average response. So you can do This is still done. We are getting to the point where we can understand most of the dynamics that happens in this course of nanoparticles. Some of this work we are doing. Now we have fresh data that we took this summer in the lab of Manuel Anzola Portoles. But again, not ideal. And there's also another push, very recent, for measuring not the optical response, but measuring photoelectrons. So this is work from 2023, where it is also a kind of a palm probe spectroscopy. But the first pulse is going to excite the system, is going to pump the excitation, but the second one is going to ionize it. So they have here a nanoparticle in vacuum. They come in with an excitation pulse. They excite the plasmon, and then they ionize it. And when you ionize it, you have the advantage that you have energy resolution. So if you have light of a certain energy, and you detect an electron at a certain energy, you are sure that the electron came from conservation of energy, a photon below. So this is what they get. They get an energy distribution as a function of delay, and this energy resolution gives you the ability to probe complex systems. This is a very complicated experiment because there has to be one nanoparticle, it has to be sitting in a vacuum, so it's still far away from probing plexitons, but it is pushing in the direction of energy resolution. And if you squint, this looks like, I always say that this looks like a molecule in a conduction lab. This is actually the energy landscape for helium, where you have ground states and you have metastable states, and you have continua with different angular momenta, but in general, in principle, it works the same. So the idea is, you want to imagine doing spectroscopy on a system that you excite with an attosecond XUV pulse, and you take an electron out. So this will be you pump the system, you create an electron wave packet, then you want to understand the wave packet. And the idea is that if you only detect the kinetic energy, the most information that you can get out of the system is the density matrix. So you need to do quantum state tomography on this electron. In case, for example, that you have two kinds of gases in your, or two kinds of internal states in your gas, and you have a mixed density matrix. So that is the motivation that I explained very, very briefly. There are 3 papers that now have appeared on this topic. And the idea is you do a pump-probe experiment. You first come in with an XUV pulse, you pump it, you create an electron wave packet. This is an ionized state. And then you wait some time, some delay, and then you probe it with some infrared pulse. And the drive is that not only do you want to measure if there is an excitation or not, but you want to measure if the states of your wave packet are coherent or not. So for this, we are developing several flavors of a quantum state tomography protocol. These are 2-dimensional maps that use the time frequency Fourier transform. And so they get an additional energy access from a time delay between the pump and the probe. And we developed something called the Rainbow Kraken that uses an XUV pulse and 2 infrared pulses to reconstruct the density matrix continuously. So this I'm explaining very fast, but the point is that if you have, for example, helium, you expect a density matrix that has this shape, and our protocol can reconstruct this density matrix. So this is done for attoseconds for ionization of helium, but we are also starting to think how a similar protocol would apply to characterize plexitons, with the idea that yes, they are very complex, but if we add energy resolution, we can understand how the excitation is moving in the molecular and the metallic system. And this is we can achieve very high fidelities, at least theoretically. This one in particular has not been implemented experimentally yet. Conclusions, I showed you the ones about the plexitons, and I just add these ones, that we are doing experiments with all spectroscopies to understand the photoexcitation dynamics. And we're also developing new spectroscopies that will be suitable to understanding these complex, dirty, messy energy landscapes. And with this, I just want to thank all the people that participated in what I presented and the following.
Is there any questions?
Too much chemistry.
The very last technique is very exciting after you that you you did touch on this, but what are the prospects to let me put it this way, what would be the limitations to turn it into really a mixed domain coherent spectroscopy? So if generating this coherent sequence of ionization pulses like an action spectroscopy,
Yes.
would that be a limitation, or is it, do you think that the kind of electron detection limits? I mean, because if you could really map a mixed domain coherent spectrum, then that would be fantastic.
I think this experiment, I'm not sure if the people involved want to repeat it again. I think it was, it's very, very difficult experiment. If in addition you have to be worried if your molecule is burning, and yes, this I think technically I wouldn't do it like this, but what we are trying to investigate is if we add more pulses, if we can fold this into a Raman signal. It's a bit hard to explain, but we need to find another observable. I don't think that a photoelectron will be the correct observable, but I do think that the idea of having, so what gives you the probing of coherences in one scan is this strange combination of a long and a short infrared pulse. Keeping that and incorporating it into a fifth order technique. Maybe I can tell you later in detail the idea, but you need to find a photon emission method that encodes the same idea behind the rainbow kraken, not photoelectron spectroscopy for molecules.
So Daniel, I have a question. How do you construct the density matrix?
How do I construct the density matrix? In 30 seconds, I have it here. So sorry. I mean, it is a bit, yeah, I think here. So it's this, no. Okay, it's this very small part. So what you measure is a photoelectron spectrum, and you measure it at different delays. When you measure it at different delays, you encode, basically you encode phases that when you Fourier transform them, they give you information on the energy levels between your photoelectron wave packets. So you get this is what you would measure, in fact. Or rather, you measure an interferogram, you Fourier transform, you stretch, you invert, you hermitize, you project into the phase of real density matrix, and then you get this. So it's complicated. It's a bit complicated, but it works very well.
Thank you very much. So I have a question. You have this mixture of polaritons and electrons, at some point of your talk. Can you comment on what's the density of the typical densities of both the polaritons and the electrons you are creating in this mixture?
I haven't measured it, but for people that do measure charge transfer, I think they can put up to 50 charges inside a nanoparticle.
Okay, so
That was your question.
can you regard I'm just thinking it's like electrons on top of a reservoir of polaritons, or it's the vice versa, or it's just more like
No, what happens, let me just go the way, but if you go, for example, here, what ends up happening is that you excite your polariton branches, and then the polariton branches decay. So you're used to decaying to dark states in the molecule, but in the metal, it's like Landau damping. So you do Landau damping, but instead of the plasmon transition decaying, the polariton states also decay, and they create hot electron hole. They create hot electrons. And then the excitation that is remaining here can just hop onto the surface. But I don't have an idea of what's the ratio. The experiments that we are now doing is measuring the transient absorption at different light intensities. And it's well known what is the behavior of an excited metal depending on the electron densities in your non-equilibrium Fermi distribution. And so we are comparing that with what you get from the plexitons, but we haven't finished the analysis.
Is there any other question?
As I understand, the suggest geometry is curved, right?
Yeah, there is a curve in some of the sulfonate chains that are probably not observed on the surface.
And what does the image learning conclusions mean? That image?
This one?
Uh-huh.
I mean, this one, okay, so what does it mean? Your question is what does it mean, or what is the...
Yeah.
It means, so with Raman and with NMR, we can see this curvature based on the dipolarly observed proximity of the spins. When this one just means that some of the sulfonate groups are going to be hanging out. So there's a surface, it's covered in plexitons. Sorry, there's a surface, it's covered in these cyanic dyes, and there are these sulfonate groups dangling. It just means that. Right now it doesn't mean anything else, but we have 3 or 4 candidate molecules that we want to incorporate along with this one, and we don't know what will happen. So we need to see if it will form like something called hetero-J aggregates. And so you form band structures, but they can come from different electronic states, and we don't know. We don't know what are the possibilities of mixing them with this TPPC. It's a lot of unknowns, but at least we know what it looks like. That's what I would say.
Thank you.
I think there are no further questions, right?